Hello everyone, we are from Group 2 and we are going to present our review paper which is about recent advancement of MSG upstream production and downstream processing. MSG or monosodium glutamate is a food taste enhancer and food additive. It's a derivation of glutamic acid founded by Carl Ritthausen in 1866. Ever since its inception, there is an increasing demand for MSG, which means the higher need of advancement in MSG production technology. That's why we wrote this review paper, as we aim to understand the growth of the MSG industry by analyzing the bioprocessing methods of MSG production.
Following the introduction, we will now elaborate the upstream process of MSG. The most important aspect of upstream processing is the microbial strain selection. From the table, we see that C-glutamicum produces highest production yield at 80 gram per liter. Notable advancements in microbial research has focused on the study of mutants and genetic engineering to optimize production. Furthermore, studies has begun to investigate the prospects of immobilization due to its practicality. Another important aspect of fermentation is the feedstock and nutrient. We know that glucose is preferred and can be sourced from both edible and inedible biomass. However, nitrogen source, biotin content, and supplementary substances are also important to monitor to optimize the fermentation.
The third section of upstream processing discusses about the glucose extraction for MSG production, which includes the most common substrate for extracting glucose, the methods, and improvements of methods. Next, for the fermentation process, this paper is adapted from Abdenaizer et al. In 2021, which compares the yield from batch fermentation and fed batch fermentation. The batch fermentation was previously used in fermentation of MSG, but then left behind due to inefficiency for the strain to grow optimally, which results in a low yield of glutamic acid.
The more recent method is the fed batch technique. This technique is better than the previous method as it allows for continuous nutrient feeding to the cells inside the bioreactor. The differentiation of this technique is from the timing of which nutrients are fed into the bioreactor, which have interesting results which I will cover later on. They both use date juice as the material with the same amount of total sugar concentration. The table shows a comparison of the three fermentation techniques, which shows that with the same value of substrate used, the fed batch technique came up more efficient than the batch method based on the value of the titer. The continuous feed has slightly higher values of titer, yield, and productivity than the pulse feed. However, the lesser biomass value on the pulse feed can be considered superior for the long run, reducing the complexity during downstream process. Lastly, to obtain an optimal yield, pH, temperature, agitation, and aeration should be maintained around the values stated here throughout the fermentation process. Now let's talk about the downstream process. The first process is the glutamic acid extraction. In the early days, GA extraction is done by using aqueous extraction, which is complicated and dangerous for humans, as it contains many impurities. Nowadays, GA extraction is done by concentrating the fermented broth to separate the broth from contaminated condensate.
MSG is refined through decolorization and crystallization. After the last process, decolorization is performed to remove the dark brown coloring. Crystallization is then performed to get the final coarse powder consistency. On that note, the MSG purity has increased from 78% to 99% through a series of advancements, as has been mentioned previously.
The utilization of tail liquid for fertilizer using spray granulation technique reduces the amount of compounds available in the wastewater, which in return drastically reduces the wastewater amount and chemical oxygen demand compound per ton of glutamic acid produced.
In conclusion, the technological advancement in MSG production process brought many benefits. In upstream process, the usage of C-glutamicum is proven more efficient than E. coli. Also, the usage of optimum parameters helps significantly. And then in the downstream process, the purification using activated carbon and multiple stages of crystallization have shown significant increase in purity of MSG and the reduction of wastewater. We also have some recommendation for future research. We recommend there should be future research into mixed media for fermentation as well as improvements in better parameters and monitoring technology. And these are our references, and thank you for watching.